Yeah, I've been involved in the potato industry all my working career, predominantly involved in seed, and then latterly I've taken over as a secretariat of the British Potato Trade Association. So Ian Toth asked me whether I would be prepared to give a talk on the potato sector within this forum. So anyway, here we go. The title I put together was the risk of bacterial pathogens to the potato sector and how science is helping reduce these risks, because I gather that they were looking at trying to involve more stakeholder presence and guide research, etc. Anyway, but from a potato point of view, there's 5 main bacterial pathogens, which on the screen there. Common scab and blackleg are the 2 major ones in this country that we've effectively learnt to live with. The following 3, brown rot, ring rot and zebra chip, they are present to varying extents around the world, some in greater presence than others, but at the moment so far we've managed to pretty much effectively keep them out of the UK, and interestingly they're all those 3 are quarantine organisms, or classified as quarantine organisms. Brown rot, it is one of its concerns for establishment is that it does have, like many bacteria, have alternative host in Solanum dulcamara, which is present in a lot of watercourses within Europe, and therefore it survives whilst outside of the potato host. The risk, therefore, then for the potato sector is from irrigation out of those watercourses onto potato crops, and then cross-contaminating into the potatoes. So the control mechanisms at the moment are predominantly based upon the regulatory, it's a quarantine organism, and also annual surveys. Now, those annual surveys are not only time consuming, relatively difficult to do, because you've actually got to get into the watercourses to get the samples. So one of the potential that science may well be able to help in the survey of brown rot would be if they can do any remote sensing of infected Solanum dulcamara. Now, whether that's going to be possible going forward, I don't know. I gather there's good moves towards using different areas of the light spectrum to detect disease plants within crops. So whether it's sensitive enough to do that within plants within a watercourse, I don't know. But that's one area that I think will be helpful. Ring rot as a bacteria, and I note there's several bacteriologists within the audience, so they'll know far more about it than I do, but it's extremely well, it's quite a acute bacteria in that it survives as a latent within the potato tuber, so it can move over several generations before it actually shows itself, and by which time it's already quite widely spread within an area. So the other thing is, I don't know how usual this is for bacteria, but certainly ring rot can, I'm told it can survive as a dried slime on machinery and on containers and on boxes. So, and likewise, it can then rehydrate and then start an infection again. Within the UK, there was no recorded outbreaks until 2003. And then that outbreak occurred then. It proved to be extremely costly to the individual on whose premises the ring rot was found. I think various figures come to mind, but it cost that individual anything between £500,000 and £1 million. So anything we can do to keep that out, or and it also triggered the or re-triggered the interest in insurance schemes for quarantine or bacterial organisms. As far as risk reduction is concerned, what science can do to help on that, other than being able to more accurately detect it, I'm not too sure. But the other one, zebra chip, that was only relatively recently identified in Mexico, and despite New Zealand's renowned biosecurity measures, it found its way into New Zealand in May 2008, or probably prior to that. It was identified in May 2008, and allegedly got in there via another host plant. So it's and cost the potato sector in New Zealand, and is now costing the potato sector in the US considerable sums of money, because the picture on the right there shows you what happens to potato crisps when they're infected with this particular bacterium. So again, for a risk reduction, the ability to be able to detect the vector on alternative hosts such as fine tomatoes would be pretty useful. Common scab, that's global, and we've learned to live with it. And the controls really are varietal resistance, and also controlling the environment to make it less suitable for its multiplication. In this case, it's through the use of irrigation. The downside of that is, by using irrigation, you increase the prevalence of another fungal pathogen called powdery scab, and also another bacterial pathogen, blackleg. So whether, from a science point of view, whether we can better understand the resistance mechanism within cultivars that do show the resistance, and then hopefully enable more rapid selection during plant breeding through molecular selection of new varieties. Blackleg, which was what I wanted to concentrate a little bit more on, I think is quite a good example of science assisting industry, and industry assisting science. It was first identified in the 1800s, and fairly rapidly noted that the disease, or expression of the disease, was associated with seed, and so to try and control it, they started using seed treatments in the early 1900s with formaldehyde, and they found that it dramatically reduced the incidence in daughter crops. And also, as a spin-off from that, it also helped instigate the use of certification or seed selection process, classification schemes, etc. The picture there gives you an indication as to what can happen in the field when you've got seed potatoes that are either heavily infected with blackleg, or find themselves in the environmental conditions where blackleg will express itself. As you can see there, the rows are fairly wet. One question that comes to mind is, why is it you get some plants that seem to remain healthy and other plants just either rot off completely? So that one reason for that is that they found out that the rotting of a plant or a tuber is directly related to the level of bacteria found within that seed tuber, and also the environmental conditions under which that tuber or which that plant finds itself. So that led to a risk matrix along the lines of this graph here, which from HDB put together following work done in the 1980s. What they would consider high risk is levels above 10 to the 5, if you like. Intermediate was levels between 10 to the 2, 10 to the 5, and low was below 10 to the 2. So I think somebody obviously well aware of the quorum sensing mechanisms that are that appear in a number of bacteria, and I think it's obviously related to that. One of the problems that cropped up from this is that when people sent samples in for testing, they would come back with quite a wide range of levels of bacteria on the different subsamples, and also they found that the resultant crops often bore little relationship to the levels of blackleg that were found when they sent the seed in for the test. So it's not necessarily been used as much as perhaps it could have been. So that there gives you a picture of what it does to the tuber, and then that gives you an idea of the result of what happens when it goes wrong. And again, within that store there, you can see there's obviously been a spot within there that's had the correct environmental conditions for the bacteria to multiply and result in a small store collapse. There's quite a lot of other research that's gone on over the years, partly industry backed, partly backed by governments. In the 80s, they looked at the hot water treatment of seed, which worked, certainly from an experimental point of view, worked very well. But from a practical point of view, dipping the seed in hot water up to around about 50 degrees centigrade, it not only killed the bacteria, in some cases it killed the growing sprout as well. So it never really got off the ground commercially. Chemical seed treatments, I think going back even further, the 1960s, I think there were some mercury treatments used, but again, whilst they worked fairly well, there were environmental issues, whether that was health and safety or whether it was the fact that it was killing the salmon in the rivers in Scotland and upsetting the fishermen. I'm not too sure, but anyway. Over recent years, AHDB have put out fact sheets. As one of the previous speakers said, one of the big issues is whilst we know information, we know what we need to do from a science sector, it's actually getting the message to the growers and the people that need to know the message. So the AHDB fact sheet has been quite a useful resource, and I think if I can see if I can find that's a sheet put together with between the AHDB and Scottish Government, and it gives you quite a lot of useful information. Some of it's just pure factual information, and the idea behind it was that if the growers and people in industry have an idea of what is causing the problems, or why they are getting the problems, they can look at potential solutions. So if you get onto the control there, you've got it gives you a good indication as to how you should be handling the material, and for example, not creating condensation moments, because when you do, you end up getting a multiplication of the bacteria. So let me go back again.
We have 1 more minute, if that's all right, Gerard, just to finish off.
Yeah.
Thank you.
And I think within the project that's ongoing at the moment, also looking at a decision support tool, again to help people, to help growers. And with that is the recent understanding that blackleg is closely related, or there's a close relationship between free-living nematodes, which potentially may act as vectors. So that gives you a brief outline of some of the work that's ongoing at the moment. And then for the future, they're looking at potential ways of identifying plants, whether you can identify those plants using spectrometry and then perhaps spot spray them out, or introduce naturally resistant genes from other closely related species, or perhaps the issue may be solved if we go down the true seed route rather than seed tubers. So it's plenty of research and plenty of opportunities. Anyway, thanks very much.